Hello. So very good afternoon. So in this session, I will be talking about the various platforms which are available for the quantification of the library. So once you have your libraries in your hand, there are two important things. It's quality and the quantity. Quality generally we are accessing using the Bioanalyzer. You might be knowing, okay? And for quantification is very crucial step because you might be knowing that while setting up the Illumina run, the initial amount of the library is in terms of the nanogram per microliter. But in the ultimately in the flow cell, you have loaded only 10 to 12 picomole in terms of the picomole. The quantification again, why it is necessary? Because it will, if we talk about the Illumina, it may lead to the under clustering or the over clustering. And in case of this ion tolerant platform, it may lead to the polyclonal. So one method is fluorometry. So generally, Qubit fluorometer is widely used for the quantification of the NGS libraries, and it is more sensitive than the UV, like the nanodrop or the QIAcuity systems. They are using the 260 by 280 ratio or the measuring the absorbance at the 260 nanometer. Why it is accurately? Because it is using the fluorescence level probes, and that are specifically bind to the DNA only. And for RNA quantification, it will bind to the single stranded RNA only, and that's why it is specific. This fast and reliable. Within 3 seconds, it will give you the result once you put your sample inside the machine. And it use as little as only 1 microliter of the sample is required for the quantification. So various kits are available for the qubit quantification. Sorry. Okay. So various kits are available for the qubit, like the DNA broad-end assay kit. The quantitation range is 2 to 1000 nanograms. Similarly, qubit DNHS kit, high sensitivity assay kit, generally for the quantification of the library, we will be using this kit, HS kit. Quantification range is 0.2 nanogram to 100 nanogram. Likewise, for the single stranded DNA, there is also kit for the RNA quantification. RNA again, RNA-BR and RNA-HS kit, and for the microRNA quantification. Second is Bioanalyzer. We are using Bioanalyzer for the quality or the to see the profile of the library. But simultaneously, a Bioanalyzer will also give the concentration of each peak available in the electrophotogram. And in a summary, it will also give you the average base space, average size of your library, and the concentration in terms of the picogram per microliter. So one method is Qubit fluorometer. Second, you can use this bioanalyzer concentration. Same, it goes for the DNA-HS kit for the library, and similarly, you can also measure the concentration of as well as the quality of the RNA using the RNA nano kit in the bioanalyzer only. Here it will give you the concentration of the RNA. You can also use these kits also. This one, RNA-HS or the DNA-HS kit for the bioanalyzer that the concentration. Then the third method is either real-time PCR or the digital PCR method that we are currently using. And for the ion torrent platform, the ion library quantitation TACMan kit is available, as well as the ion universal library quantitation kit is available. They both are using the TACMan chemistry. And for the Illumina platform, we are using the kappa library quantitation kit. It is using the cyber chemistry. So how I think I have already discussed in the wet lab session why quantitation using these kits like a digital PCR or real time PCR is more accurate as compared to that Qubit fluorometer or other methods. Because say for example here two different fragments of libraries are there. So if you quantify this library using the Qubit fluorometer, it will give the concentration of both of these. But if you quantify your library using either real time or the digital PCR, which is using the adapter specific primers, then it will quantify the library which is having the adapter sequences. Because in library preparation, it may happen that old DNA fragment may not have the adapter sequences. It will not going to be sequenced. Same is for the digital PCR. The advantage of digital PCR is that no dependency on the standard of the reference, because in real-time PCR we need to run standard curve each time. However, here it is a absolute quantification without the need of the reference. And the linear detection of the minute fold changes, because a single copy can be also detected by this machine. And a highly inhibitor tolerant, because here the in real time PCR, what will happen? The all the reaction will be happening in the one PCR tubes or the one PCR tube well, 96 well plate. Here your sample will be distributed like the emulsion PCR. It will be partitioned. So there will be a chance of the chance of getting that PCR failure is very less as compared to the real time PCR. रियल टाइम पीसीआर में रिएक्शन इनहिबिटर होगा तो पूरा रिएक्शन फेल हो जाएगा. यहाँ पे क्या होगा कि हर एक को पार्टीशन में डिवाइड हो जाएगा. सभी पार्टीशन में इनहिबिटर जाएगा वो पॉसिबिलिटी नहीं है, तो चांसेस ऑफ दैट पीसीआर इनहिबिशन इज वेरी लेस. These are the comparison of this traditional versus the digital PCR. That is, it measures the number of the target molecule directly by the counting the positive fluorescence well directly from the well or the partitions. And digital PCR allow the absolute quantification of the target molecule using the unique blend of the sample dilution and the Poisson statistic. And it is highly sensitive and accurate and can be used effectively in the absolute measurement of the nucleic acid or the gene detection, copy number detection, etc. and it provides the user-friendly result from in the digital form. You will directly get the copy number for in your sample. Thank you. Now we will be having a lecture on the digital PCR. Not there.
Good afternoon, everyone. As discussed by Dr. Ramesh, the brief about the digital PCR. So, here we are discussing about the principle, how digital PCR actually works, and what are the applications which can be performed by using the digital PCR. So, digital PCR is a common term which can be used by different instruments, uh, different PCR systems. So that can be formed based on different kind of the principles of partitioning. Suppose the digital PCR systems from the BioRed, which is having the principle based on the droplet formation. Then from the Kaizen, digital PCR is based on the nanoplate based partitioning method. And applied biosystem is also having the digital PCR, which is based on the chip based partitioning. So digital PCR is a system. It is a third generation most advanced PCR technology, which is designed for specific detection and absolute quantification of nucleic acid, can be a DNA or RNA. So here, what is the beautifulness of this system? It can be work based on the real time PCR chemistry, as well as the workflow is also similar to the real time PCR. And here also you can use the reagent like Primaprobe, what you have designed for the real-time PCR can be used here. And the master mix composition can be also the similar, but sometimes you need to use the proprietary item, proprietary consumables for the digital PCR use. And here, compared to the real-time PCR will give you the actual real-time fluorescence or amplification as the cycle progresses. But here, what you will have is a first you put the sample, it will partition it, it will amplify, and then you have a detection. So it is called as an endpoint detection. And here, as mentioned by Dr. Ramesh, it is absolute quantification without a need of any reference. And why it is a digital? Because it is depends on the binary signal, 1 or 0. So your PCR reagent will be distributed and having either 0 copies or 1 copies. And the major advantages of this digital PCR is it quantifies the low abundant target molecule. So firstly, in 1983, the conventional PCR was evolved. Then in 1996, the quantitative RT-PCR based PCR came across, and 2006, the digital PCR came in the market. And now, on the basis of digital PCR, it can be through any company, but over 3,000 digital PCR based scientific publication has already published, and it is still counting. So let's compare these three generation PCR methods, PCR technology. So first one, conventional PCR is basically the qualitative. You will not get any idea about the concentration or the actual copies, and it is a technically simple and it is a very low cost method. And here also you will see the result at the endpoint. So it is an endpoint detection. The relative quantification by the real-time PCR is compared to the compared to the conventional PCR, it is having higher accuracy, sensitivity, and specificity. And it is also will give you the picture at a real time manner. And the digital PCR is based on the absolute quantification, and it is also giving you the high precision as well as reproducibility. And it will have a low sensitivity to the inhibitors. So your PCR inhibition will be quite low compared to the other PCR technologies. And as compared to the conventional PCR, it is also having the endpoint detection method. So mainly the digital PCR, how digital PCR works. So all the phenomenon, how the digital PCR work is based on the partitioning. So partitioning can be through any way, through the droplet generation, through the channel, or through the any way. So firstly, you prepare your sample. It can be genomic DNA, can be RNA, or can be your cDNA, and then your sample will partitioning of your sample will be there in different manner. Like if you are using 26,000 partition well displayed, then it will be partitioning into the 26,000. And if you are using lower and suppose biorate droplet PCR is having 20,000 partition based on the droplet. So whatever number of partitions are there, your reaction mixture will be evenly, it will be distributed, and then at each separate partition, your PCR will amplify, and then final readout will be your positive partitions as well as negative partitions. So positive partitions will have your amplification, and negative partition will not have any amplification. So what are the several advantages of digital PCR over the real-time PCR? So let's see the precision. So the precision will be very low in the real-time PCR compared to the digital PCR. If you perform the experiment for mutation detection, so the mutation detection rate using the RT-PCR will be the greater than 1%, and here it is very low. The detection here, the major advantage of digital PCR is that it detects the small fold changes and rare targets, and it plus or minus having the 10% of precision. And here, yes, it requires a standard curve. Here, it not required any standard curve. PCR inhibitors tolerance is different. Here, if you have PCR inhibitors present in your reaction and performing the RT-PCR, so if PCR inhibitors are there, your PCR reaction will not go and it will be inhibited. And here, because it is dividing into so many minute reactions, so your inhibitors PCR tolerance to the PCR inhibitors is very higher. And if you talk about the reproducibility, so it is higher in digital PCR. But yes, real-time PCR is less expensive than the digital PCR. So as I said that here you can detect a small difference in your gene expression as well as in the copy detection. So suppose if you talk about the standard curve here performed by the real-time PCR, here suppose you have a one cycle, one CT change. So you have the fold change. Suppose here, so in the real-time PCR, suppose you have a 3.3 CT difference, you have a 10-fold changes. But here over the one cycle threshold, you are having this many of the points where you can detect small changes. Suppose this is a one cycle threshold here from the real-time PCR, but here you can suppose it is a very minute difference into the copies. Then also you can detect. So it is a very dynamic range for the detection. So what are the mainly? So it is done in a 4 different steps. Firstly, you need to do the sample dilution. As I also mentioned in the practical session that suppose first time if you are doing, then you need to do the dilution titration to give the sufficient amount of your positive and negative partitions. So you do the sample dilution for over concentrated samples. If you will start with the over concentrated sample or higher concentration of the samples, you will get all partitions positive. So it will be oversaturated. And number of negative partition is also required. Some number of negative partitions is also required for the accuracy. And here also, like the RT-PCR, it can work with both. That is a intercalatic type, EVA green, or it is a probe-based reaction also. And then you make your PCR reaction using master mix, primer probe, and your sample, and then you load into the digital PCR plate. So it can be through the droplet, but here we have a QIAgen based QIAcuity digital PCR. So it will work with the partition generation. So we have two kinds of a digital PCR plate, 8.5 thousand partition plate as well as 26,000 partition plates. So whatever you load, whatever PCR reaction mix you load into the well, it will partitioning into the thousand of individual reaction, and this thousand of individual reaction will be carried out the PCR reaction for the amplification of your target. And after amplification, after completion of your all entire PCR cycle amplification, you will read the fluorescence in all the partitions, and finally you will do the data analysis. So the digital PCR is mainly based on the Poisson distribution. So let's see that what is the even versus random distribution. So if you have an even distribution, suppose you have 100 molecule and you have 100 wells. So this is a even distribution. Here all 100 wells is having one copies. So this is even distribution, and here this is a random distribution. So if you have 100 molecule and you have 100 wells, so it is not randomly distributed. So suppose here 37 wells is having not having any molecule. Others are having one molecule, two molecule, three molecule. So this is the digital PCR is working with this random distribution process. But here, suppose if you here we cannot count individually. So we need the statistics. So this digital PCR is working with the Poisson distribution statistics. So this is the distribution pattern this digital PCR is working. So suppose here the red one is a positive well, so where the fluorescence has been detected, and the gray one is a negative. So there is no fluorescence in that well. So it can be anything, like some wells is not having any amplification, some wells are having more than one. So according to that, the 95% of confidence interval will be counted. So this is the suppose what I told, that you need to first titrate the dilution of your starting DNA or RNA. Suppose if you are not having any amplification, so your all partition will be negative. If you have a few copies present in your target sample, so this is a lower end signal and this is a higher end signal. So these two are good for counting the final copies in the reaction. This is oversaturated. Suppose you are starting with very high concentration of RNA, cDNA or genomic DNA. Your all the partition will get saturated. So it should not be there. So you need to select the dilution according to the saturation of your partitions. So from this, statistically software will count the copies of the DNA or RNA target per microliter basis. So as I told also, this is oversaturated. This is the valid saturation, valid partitions, and this is a no amplification. So your NTC should be like this. So from these kind of patterns, the software will calculate the concentration. So like the RT-PCR, it can be two kinds of the chemistry can be used. First is a Eva-Green based, and second one is a hydrolysis probe based chemistry. So as you all know that this is Eva-Green is an intercalating dye with a double strand binding affinity. So the green channel what they have provided in this QIAgen digital PCR system will work with this Eva-Green based chemistry, and here also like the cyber-green, whenever you do the cyber-green based PCR reaction into the RT-PCR, you will get non-specific PCR products also get amplified. So here also the same thing, but here what additional advantages is there, that suppose if you have any primer dimer, it will give you the fluorescence signal, but it might appear as a separate group when you analyze your results. And this is a Techman probe based detection. So here your fluorophore is as a fluorophore and quencher has been there at the both end of your template DNA, and during the extinction phase, this fluorescence due to the 5' to 3' exonuclease activity of your DNA polymerase will break down this fluorophore, and it will be detected by the filters, and it will give you the fluorescence. So the digital PCR system from the Kaizen, what we have is coming with the two different kinds of formats. So they are also giving two channel systems. So that will be work one with a EVA green and one with a FAM based technology. But what we have here is a 5 channel system of digital PCR. So we have this all 5 different channels which will work with different excitation and emission wavelength. And from this is the example fluorophore they have given from the specified filters. So these kind of dyes we can choose for your multiplexing protocols. But this is the example dye they have given in the software. But suppose any dye which is not mentioned here, let's say suppose Psi3 or AVY, we can also select that fluorophore also, and we can select the emission wavelength, and we can use this yellow channel or red channel, whatever is coming for that particular dye. So how it calculates the copy number? So here the total number, because whenever you do the digital PCR, your result will be finally it will be the what are your positive partition, what are your valid partition, what are your positive partition, and what are your negative partition. So your valid partition is that whatever reaction mix you load into the well, that will be distributed into the 26,000 partitions. So all partition will not have your reaction mix. So suppose from the 26,000 partition, if your 25,000 partitions are having the reaction mix, that will be called as a valid partition. So that only partition will be considered for further calculation of copies. So from that valid partition, how many are positive partitions, which are giving the fluorescence, and how many are negative partitions. So the total number of copies of target molecule will be based on this. So this formula, suppose this is a log of number of valid partitions minus number of your positive partitions divided by number of your valid partitions. Let's say suppose 8,000 is your valid partitions which are having reaction mix. From that 4,000 partitions are positive and 4,000 partitions are negative. So you will calculate this. So this lambda is your 0.693 based on this. So here 0.693 you need to multiply with your total valid partitions. So that will give you the copies. That will give you the copies, and here on base because whatever volume you are adding to the PCR well, that will also determine the copies. So this is the volume, the delta divided by volume in a microliter. So let's say your delta was 0.693 based on this, and your estimated partition volume is this, because according to the number of partition and whatever total volume you are applying, so how it calculates the final copies in the software. So this is the formula given by the manufacturer. So now, what are the limits of the detection? Suppose the two plate formats are coming with the digital PCR, 8.5 thousand partitions and 26,000 partitions. So in this, around 8,500 partitions will be there. Here 26,000 partitions will be there. So as in practical also, suppose we have used this 26,000 partition plate, and there we loaded 40 microliter of sample. So from this 40 microliter, your analyzable volume is this much only. So from 40 microliter, analyzable volume for this is a 24 microliter, here approximately around 3. So uh, per partitions, it can detect 5 copies with the probe-based assay and 2 copies for the EVA-green-based assay. So let's say after calculating, this will be calculated by the software, like what are your values, as I mentioned earlier. So 42,500 copies, whatever volume you are loading, can be detected in a final output. So up to this, you don't need any kind of dilutions. But if you are going beyond this, it will get oversaturated, and then you need the dilutions. So this is the limit given by the manufacturer for both kind of well formats. So this is the what are the process parameters which are changing in both the method RT-PCR and digital. So it is a very similar. Suppose if you take the Bioanalyzer droplet-based PCR, so their workflow is quite different. Firstly, you need to, that is based on the oil in water emulsion-based droplet generation. So firstly, you need to make the reaction mix with the your sample and your oil in water, and then your droplet will be generated in a 8-well cartridge. So individually 8-8 well you need to generate the droplet, and then separately you are putting into the PCR well these generated droplets from your PCR master mix you will add into the PCR plate, and then so this is a 3 different kind of system they are provided. But here Kaizen, the workflow is very similar to the RT-PCR. Here the like same, we are making the mixture and then we are loading into the plate and then we are analyzing. So this is the instrument I think you must have seen. So here at the above side, this is the imaging or readout module. So whenever, so from here the plate will come out and you will place your plate after sealing. Then this is the screen and your partitioning and everything will be here. Partitioning, rolling and all other modules are here. So thermal cycler will be here and your imaging will be here. So this is the like plate format. So this is a just a representative of the wells. So this is, let's say in 24 well it will be 24. This is 96 well. So this is a well. So this is a zooming of that particular one well. So this is your input well. Here you are putting your samples. So your samples will be over here, and this is a small 26,000 channels. And extra volume will be there for in the vent. So this is a cross section view of that particular well. So what are this is a nanoplate base as well as droplet base. So we have a nanoplate base. So these have several some advantages, like whatever partitions is there that will be sealed from the bottom when you put the plate inside the instrument. So sealed partition prevent the variation in the size, as well as it will avoid the well-to-well contamination also, and faster readout compared to the other technologies. And this is a simple workflow. So whoever is handling the RT-PCR, they can handle this system. And this is a, so firstly, you make a reaction mix, put here. So this is your sample having well, where having your samples. So here, what the piston from the above side inside your instrument will come and it will push your liquid from the well to all these channels. So what we tell valid partition is when your plunger is pushing your liquid, so even ideally all wells should have your reaction mix, but some will not have. So according to that, they are counting the valid partitions. So after distribution of your PCR mix from one well to all the channels, the roller from the bottom side inside the instrument will roll out and it will seal all your micro partitions. So your all your these partitions will act as a different wells. So it will act as a separate 26,000 wells, and it will amplify as a separate reaction. It will give you the fluorescence. So it will give you the higher sensitivity. So these are all 3 formats. So this is 24 well, 26,000 partition plates, which we have used in the practical. This is a 8.5 thousand partition plate having 24 wells, and this is a 8.5 thousand partition plate having 96 wells. So these are the some of the difference. So here 40 microliter input samples they are recommending. For this they are recommending 12 microliter for 8.5 thousand partitions. So different applications. Suppose your target is very rare, so you are advised to use the 26,000 partitions, because as many as partitions are there, more partitions are there, you will have a more amplification and you have a higher sensitivity. But if your target is very abundant, then you can use 8.5 thousand partitions as well as. So here main reference channel is there. So whenever you do the analysis of your results after completion of the digital PCR, you will see the reference channel. So here they will show like this. So it will count the number of field partitions, and it will determine the analyzable volume. Suppose your partitions is your valid partitions are less, so your analyzer volume may be also less, and your result will not be that much good. So, and your green channel will give you the your target fluorescence. So, let's say, as I told in the practical also, suppose if you are having bubble or anything in your reaction mix, where while you are loading into your well, digital PCR well, then in this 26,000 plates, they are having four kind of four channels you can see from this one well. So suppose if you are having bubble, then your ideally your all four well should have clear reference channel without any interference. But if you are having the bubbles, it will show you the blockage of any of your portion of your that partition. So you will clearly see into the results. Suppose if you are finding that only 14,000 partitions are valid, and if you go with this reference channel, you will see that yes, some portion are not detected due to bubble or any other error. So this is the software setup. I think you must have gone through this in the practical session. So if you have any query, we can go over there in front of instrument, and you can ask anything regarding the software or regarding the analysis. So here basically you need to select which kind of plates you are using, what is the thermal cycling profiling, and you can give the different name to the reaction mix. Suppose I want to do the multiplexing. Suppose out of 24 wells, my 5 wells are I want to target for FEM. Other 2 are having sci-fi, or other 3 are having Texas red. So I will make the reaction mix, and I will give that channel or that dye particular for that reaction mix, and then we'll assign the samples. And then the normal cycling profile, what you are doing in the RT-PCR, you need to set up. You need to set up the samples and control, and then you start your run. And whenever your run is completed, then you see like this your results. So they will also give you the different inbuilt calculation for absolute quantification, mutation detection, or copy number variation kind of applications. So these are the results parameters which has been given by the softwares. Firstly, you will get the plate image, then it will a histogram for your fluorescence signals. This is 1D scatterplot. So here, what are your negative partition and positive partitions? 2D scatterplot is used when you are using different multiplexing options. And this is a signal map. We are, what are the signal like? How is your signal? It is oversaturated or it is very low. This is a heat map which is giving the concentration per partitions. This is a point diagram, and they are also giving the concentration diagram for all the wells as well as for all the targets. So what are the different applications we can use? We can perform using the digital PCR, so mutation detection, copy number variation, gene expression, miRNA detection, gene editing, SNP genotyping, or any viral or pathogen or microbiome analysis, uh, NGS library quantification, NGS validation. So this is one example of copy number variation. So how they perform? Suppose, so this is a, they want to check the aneuploidy using the digital PCR. So the non-copy number variation contains, this is a XY, so it contains the one copy of X. This is XS that contain two copies, and triple X is a three copies on the X chromosome. And human beta actin gene they are taken as a reference to normalize the gene copy number. Here you see that in the NTC they are not ideally it should not give any positive partitions and or any signals, so it is not having any one. So suppose here 1x is there, so your their final detection is 61 copies per microliter, and here double x is there. So it is, if you consider these two, it will have a two-fold increase in the copy number of that is specific for x, and here in the triple X, it is having threefold. So it clearly indicates these conditions. And here the same for the Y chromosome specific. Here one is there, and here no Y chromosome is there. So they are not detecting anything. So likewise, you can design your experiment for different application, and the Kaizen the manufacturer is also giving the customized assay for different application that is compatible for this digital PCR assays. Do you have any questions? When we analyze the results, anything you want to add?
No, it's fine. Perfectly fine.
So if you have any queries regarding the analysis or when we go there, so I'm there only, so you can ask.
देखो लैपटॉप बंद होगा